Good evening, one and all. I warmly welcome you to our CellTalk webinar. In today's session, Dr. C.L. Bharti will be presenting research titled Expression of CD133 Cancer Stem Cell Marker in IDH-mutant and IDH-wild type Isocitrate dehydrogenase Astrocytoma. Dr. Bharti is currently working as a clinical content ambassador at Renix Life Sciences. She is a dentist whose areas of specialization include relief of immediate tooth pain. She completed her MDS from Krishnadevaraya Dental College Hospital, Bangalore in the year 2018 and her BDS, Bachelor of Dental Surgery, from Government Dental College Hospital, Vijayawada in 2013. She did her intermediate schooling at Ratnam Residential School and College, Nellore. Apart from her post graduation, she did oral implantology and BLS basic life science course. She currently has 4 publications and 2 under peer review. Due to her interest in research, she was working as a freelance writer and associated with various research projects at NTR University of Health Sciences. She strives to work hard in this fast-paced and highly competitive environment without compromising on productivity. She would also like to explore new skills and technologies in this research field to update her knowledge and improve efficiency. So without further ado, I invite Bharti to take the floor.
Yeah, thank you, Aditi, for your nice intro. Good evening, one and all. Myself, Dr. Bharti. Today, I'm presenting my second CellTalk presentation. So, the topic I selected is related to astrocytoma. So, the title of the article is Expression CD133 as a cancer stem cell marker in IDH, that is associated dehydrogenase and IDH-wild astrocytomas. The original authors of the word is Saga et al. So the topic was selected is related to astrocytoma, and the title of the journal is Asian Pacific Journal of Cancer Prevention. The impact factor is 2.154. It was calculated in 2014. After they didn't calculated the impact factor rate. So the publication year was 2022. And this research, which was conducted, has an impact on the diagnosis of different types of astrocytoma, like what is the classification in diagnosis, and how to diagnose different types of astrocytoma, and after diagnosis, what is the scope of treatment in different types of astrocytoma, that is whether you have any recurrence or not. So moving to the topic, astrocytoma, it's nothing but a brain tumor. It is the most frequent type of the brain tumor, which has a significant morbidity and fatality rate. It's commonly seen in both the adults and children. So astrocytoma, basically, it is a type of brain tumor that belongs to the group called glioma. So depending on how severely the tumor is spreading into different tissues, these gliomas are broadly categorized into several types. So several, like World Health Organization and different authors, they have different classifications in different years, and every year they're updating it with something. So astrocytoma basically contains the overgrowth of the astrocytes, which are the star-shaped cells. They are nothing but they belong to the they come under the glial cells, which are the cells that are present in the total nervous system. Astrocytes, whereas they are the star-shaped cells, and they limit only to the central nervous system. So astrocytes, basically, they are a type of glial cells which are located in the central nervous system. So astrocytoma patients, they have a bad prognosis. And when you see the diffuse gliomas, that is the gliomas that are spreading to different parts of the body, this is particularly appreciated. So coming to the general background, according to the World Health Organization, astrocytomas are classified into 4 grades. So based on the tumor advancement and the histopathological characteristics. So type 1 is the pilocytic astrocytoma, which is commonly seen in children. Type 2 is the low-grade diffuse astrocytoma, that is the astrocytoma that is spreading diffusely. But when you see the histopathological classification, it comes under the low grade. Third type is anaplastic astrocytomas, that is they have the characteristic features of anaplasia and all. And the fourth one is glioblastoma. Glioblastoma is also a type of glioma. So in the recent times, by including several molecular parameters like the isocitrate dehydrogenase mutant expression and several others, several new classifications were developed by several scientists and World Health Organization. So astrocytic tumors, they were previously grouped. So now diffuse gliomas are also included in this astrocytoma classification. Even though they doesn't belong to the origin of astrocytes and all the classifications are mixed, and gliomas are included in astrocytomas and astromas are included in gliomas. So based not only on their growth pattern, but also the expression of these molecular parameters. So these classifications created the new difficulties in testing and diagnosis of the disease. So what are all the present challenges in astrocytomas? So they are very difficult to diagnose due to their high level of heterogeneity and the complex mechanisms. And we have a need to separate the astrocytomas based on their growth pattern. That is, few astrocytomas are restricted only to certain area, whereas few astrocytomas, they have a widespread. So for that, we have few recommendations that they can be classified based on their specific gene mutations that is present. So isocitrate dehydrogenase enzyme, this is the most important enzyme that helps in the pathogenesis of astrocytoma. So isocitrate dehydrogenase enzyme, it has several types like type 1, type 2, and type 3. Type 1 is most commonly seen in cytoplasm, whereas type 2 and type 3 are seen in the mitochondrial area. So they play an important role in the metabolism of various aspects of disease. So IDH has again two types, that is 1, 2, and 3. So here we are discussing 1 and 2. They do the same function, that is the conversion of alpha ketoglutarate to hydroxyglutarate, which is the most important in the Krebs cycle and oxidation process. So it is an enzyme which plays an important role in the pathogenesis of astrocytoma. So the objective of this research paper is it evaluate the differences between the IDH1, that is the isocitrate dehydrogenase enzyme 1, and CD133 cancer stem marker expression in different types of astrocytomas. So they have taken different types of astrocytomas and they calculated the IDH1 enzyme expression and CD133 cancer stem cell expression, whether they are expressing same in these different types of astrocytomas or not. So for that, they have taken 67 paraffin block samples of patients. These patients have a they have taken three categories of astrocytomas, that is the diffuse astrocytoma, anaplastic astrocytoma, and glioblastoma. And among these 67 samples, they divided them based on these diagnostic characteristics. And these unstained slides, they were made from the paraffin blocks, and IDH1 or 132H. It's a monoclonal antibody which is used to stain this isocitrate dehydrogenase enzyme, and CD133 immunohistochemistry staining was performed. So this assessment of both these expression were assessed on the cytoplasm of the tumor cells using a light microscope. And this assessment was done by two pathologists who were blinded and they don't have any information regarding the classification of astrocytomas and classification of staining, how this staining was done. So this IDH, how it is expressing, it was divided into two groups. So first, they have seen the staining under the microscope. If the IDH1 expression was stained equal to 10% or more than 10%, it is categorized as IDH1 mutant. That is, the expression is positive, it is characterized as IDH mutant. If the staining is seen less than 10% on the membrane and cytoplasm of tumor cells, it is called IDH wild type. That is, the IDH expression is considered as negative. So next, the CD133 immunoexpression was also calculated in the same manner using the semi-quantitative analysis. They assessed the stained area based on the intensity and the percentage, and the overall score was calculated by multiplying this intensity with the percentage of the total cells. So the intensity is classified as, if it is no color, the score is given as 0. If the color is visible at 400x magnification as weak, the score is given as +1. If it is visible at 100x magnification, the score is given as moderate, that is 2. If it is seen even at 40x magnification, the intensity is declared as strong and the scoring is given as +3. So the colored area is also visible at 400x magnification, and the extensibility is calculated as, if the no area is colored, they had given 0%. If it is colored area, they have given 10%, and colored area is more like 10 to 50% and more than 50%. So the results they have tabulated based on the relationship between the two genes expressions and with the diagnosis of astrocytoma. So first, out of 67 patients, they categorized them based on the demographic variables like gender, age, location of the tumor, IDH1 positive expression and CD133 positive expression, and based on the types of astrocytomas. So among all the 67 patients, 22 patients were reported with the diagnosis of astrocytoma, and 15 patients were reported with the diagnosis of anaplastic astrocytoma, and 30 patients were reported with glioblastoma. In the gender, male patients were 40 and female patients were 27. And compared to the age, below 19 years are 9 people, and 20 to 39 years are 24 people, whereas 40 to 59 are also 24 people, and more than 60 years are 10 people. The location of the tumor, in 55 patients, it is supratentorial, that is above the temporal fossa, and in 12 people, it is infratentorial. And compared to the expression of IDH1, so that more than 10%, that is the IDH expression is positive, stained area more than 10% or equal to 10%, that is the IDH mutant. It is seen in 46 patients, and IDH expression is negative is seen in 21 patients. The CD133 high expression is seen in 33 patients and low expression is seen in 34 patients. So they differentiated the 67 patients based on these demographic variables and gene expressions and the type of astrocytoma. So next, they have differentiated based on the diagnosis of astrocytoma and the expression of this IDH1 mutant. So among the 22 patients of diffuse astrocytoma, 12 patients were given the negative expression of IDH, whereas 10 patients were given the positive expression of IDH. Among the 15 patients of anaplastic astrocytoma, 1 patient is given IDH negative expression, 14 patients were given IDH positive expression. Among the 30 patients of glioblastoma, 22 patients were given positive expression and 8 patients were given negative expression. Total among all the 67 patients of all the 3 types of astrocytomas, 46 patients were given the IDH positive expression, that is, they belong to IDH mutant type, whereas 21 patients given the IDH negative expression, that is, they are categorized under IDH wild type. So the significance levels were given less than 0.5. So these results indicate that there is a statistically significant association between the astrocytoma diagnosis and the expression of this IDH1. And they categorize the results based on the diagnosis of astrocytoma and the expression of this CD133 stem cell marker. So among 22 patients of diffuse astrocytoma, in 18 patients, the expression is low, whereas in 4 patients, the expression is high. In 15 patients of anaplastic astrocytoma, the 8 patients, the expression is low, and in the 7 patients, the expression of CD133 is high. In 30 patients of glioblastoma, 22 patients were given the high expression of CD133, whereas 8 patients were given the low expression of CD133. And the results they calculated as the probability value of less than 0.5, which indicates there is a significant relation between the diagnosis of astrocytoma and the expression of CD133 enzyme. And they have taken the relationship again between the IDH expression and CD133 expression. So IDH expression, again they categorized based on the disease wise. So diffuse astrocytoma, again IDH wild type and IDH mutant type. Anaplastic astrocytoma, IDH wild type and IDH mutant. And glioblastoma, IDH wild type and IDH mutant. So total CD3 expression was recorded, low expression was recorded in 33 patients and high expression was recorded in 34 patients. So the probability value is less than 0.5. So the IDH expression and CD133 expression are significantly interrelated when it comes to astrocytoma. So IDH1 expression and CD133 expression in 10% where the IDH expression is negative, IDH wild type, CD3 expression is probably significant, and in IDH positive expression, CD133 is also probably significant. So based on these results, they concluded that there is a significant relationship between the diagnosis of astrocytoma and the expression of IDH1 and CD133, and also IDH1 and CD133 are significantly interrelated. So coming to the discussion, Astrocytomas, they occur throughout the central nervous system, but they are usually located supratentorially, and they have a greater intrinsic tendency to develop into either anaplastic astrocytoma or into glioblastoma if the tumor spreads to diffuse wide areas. So, although astrocytoma, it's a very heterogeneous disease. This molecular classification of astrocytoma, which was done based on the expression of IDH1 and CD133, it somehow may reflect their biological behavior. That is, we have the tumors with similar expression profiles and similar gene alterations, and any signaling pathways are different or not. So, if when we are observing their biological behavior, if any mutations are identified in this IDH1 enzyme in that particular astrocytoma, we can calculate what type of astrocytoma is it. That is, whether it is a IDH mutant type or IDH wild type, regardless of its histopathological grade, like whether it belongs to grade 1 or grade 2 or grade 3 or grade 4. But based on the expression of IDH mutant and IDH wild, we can calculate whether it has a better prognosis or not. So based on we have taken to express the IDH1 and CD133, the immunohistochemical staining was performed. So this staining procedure was selected based on the results of several previous studies. They have indicated that for detection of IDH1 mutation, this immunohistochemical staining procedure accuracy ranged from 88 to 100%. And they have also stated this immunohistochemical staining is more sensitive in detecting these IDH1 mutations and compared to the DNA sequence method, especially in the tumor samples that are so small. However, these statements are contradictory to some other studies where they show that DNA sequencing is more sensitive test because this immunohistochemical staining can detect only few antibodies and other antibodies cannot be detected by this method. So coming to the CD133 stem cells. So what are stem cells? They are the self-renewing population which are pluripotent and quercetin. And even though they constitute a minor population of the cancer cells, they are mostly associated with the resistant features of astrocytoma and they help in the recurrence of astrocytoma. So these stem cells are the main reason why the patient doesn't respond to the treatments in astrocytoma and why there is a relapse in the astrocytoma. So the CD133 subpopulation of astrocytoma stem cells, they have a more malignant behavior, and with the increasing of the tumor grade, their frequency will increase. Like if the tumor grade changes from 1 to 3, 4, and the CD133 stem cells count will also increase, and they are associated with tumor recurrence. So the CD133, they are the reason for the resistance of astrocytoma to all the conventional treatment procedures that are the chemotherapy and the radiotherapy. So presence of the IDH1 mutant Isocitrate dehydrogenase enzyme, its expression can affect the presence of this cancer stem cell marker CD133. So it is proved in this study that is they both are interrelated. Like in particular type of astrocytoma, if the IDH1 expression is positive, it also showed the presence of positive CD133 enzyme. So the presence of IDH1 mutation, that is the IDH1 mutant, will increase the presence of CD133 expression. So what is the reason for this is the cell differentiation will be inhibited during the evaluation of the tumor, and whatever the IDH1 mutations that are present, they continue to recur with the increasing of the tumor grade and the aggressiveness of the tumor, so that the fraction of these differentiated tumor cells will decrease, while the cancer stem cells continue to proliferate and they will increase their population. So to conclude, the study has indicated that there is a significant difference in the expression of IDH1 monoclonal antibody expression and CD133 expression in diffuse astrocytoma, anaplastic astrocytoma, and glioblastoma. That is, these three different types of astrocytomas. So different expression of this IDH1 enzyme and CD133, and both these expressions, they have a significant relationship with increasing tumor grade. That is, when the grade increases, IDH mutant expression is changed from negative to positive, and with the increased grade, CD133 count is also increasing. So the limitations of the study, they mainly consist on methodology, but because they have taken only 3 types of astrocytomas, the other types of astrocytomas are not mentioned in the class. taking the samples, and immunohistochemical staining, what they have taken to select the gene expression. According to few studies, it is very sensitive in detecting the IDH1 enzyme, whereas according to few studies, apart from immunohistochemical studies, DNA staining is more sensitive when detecting the IDH1 enzyme. So the impact of research is it helps the researchers who are classifying the astrocytomas and who are giving the classification to differentiate different types of astrocytomas. And clinicians, if they have CD133 mutations, it indicates that the tumor will have a more chance of recurrence. So based on that, the clinicians may change their treatment plan in administering the drugs and all. And the success of the treatment plan will be evaluated based on the type of astrocytoma. Example, IDH mutant, if the IDH expression is positive, it has a better prognosis compared to IDH wild type, where the IDH expression is negative. Thank you for your patient listening. You can find me at back next.
Thank you so much, Bharti, for that informative webinar. I will now open the floor to the audience for any questions, comments, or discussions that they might have. So I see a few questions. Vijay has a question. He asks, what is the practical utility of these findings? What is the practical utility of these findings that in this paper?
So practical utility means we can see the recurrence, like whether if the CD133 expression is more in a particular type of astroma, it indicates that there might be recurrence for that type of astrocytoma. And if based on the IDH expression, we can predict the prognosis of the disease, whether it is the prognosis is good or bad.
Nomer has a question. He asks, what value does the study add to the already existing classification system of astrocytomas?
So for adding the value to the classification, it doesn't add any further information because it has also taken the classification of H4, that is the grade 1, grade 2, grade 3, and grade 4. They just I mentioned that the classification is not including the molecular parameters, and it is very confusing. So they have created a separate entity based on the expression of the CD133 and IDH1, and based on this expression, they are dividing them into like tumor with recurrence and tumor with normal. And so for the classification, it doesn't add any better value. That's what I felt.
I hope that answers your question, Nomer. Nupur has a question. She asks, is any mutation of CD133 reported?
That I don't know.
So I see no further questions in the chat box. So without further ado, I would like to thank Bharati again for this informative webinar and the audience for being here throughout. I would also like to inform you all about the next presentation titled Risk-Free WHO Grading of Astrocytoma using Convolutional Neural Networks from MRI Images. And it will be presented by Jay Darji on the 22nd of Feb 2023. Thank you everyone for your attendance today, and I would like to see you all next week at the same time. Thank you so much.